The three pillars of advanced solid cancers. Although cancer evolution is a chaotic, stochastic, iterative, and unpredictably complex process, we can try to analyze advanced solid cancers by considering the following three characteristics as its major pillars: intracellular instability, metabolic flexibility, and cytoskeletal plasticity. Following this approach, we provide a simplified overview of tumor growth and metastasis formation, starting with intracellular instability. It is well known that in addition to hereditary factors, a number of external carcinogenic events can lead to increased mutations and significant DNA damage. These events can induce strong anomalies and breaks in the DNA. On the other hand, the most frequent cause of chromosome missegregation is mitotic failure. Telomere erosion and perturbations in the DNA replication process can also lead to chromosomal deviations. Carcinogenic events can also induce various types of cellular stress. Apart from a few special cell types with polyploid structure, the vast majority of human cells are diploid. They contain 2 copies of each chromosome. To cope with internal errors and external insults, our cells are equipped with sophisticated DNA damage response mechanisms. But repair deficiencies or irreparable DNA damage can result in short-sequence nucleotide level alterations called microsatellite instability. The duplication, loss, or breakage and rearrangement of chromosomes can induce chromosomal instability, which can be further exacerbated by deficiencies in the homologous recombination and non-homologous end-joining machinery, leading to structural as well as segmental or whole number aneuploidy. These karyograms illustrate well the large chromosomal differences between a normal and a cancerous cell. Various epigenomic effects and cell-in-cell processes can also contribute to polyploidy and large-scale somatic aneuploidy. In a vicious circle of events, abnormal karyotypes can further aggravate cellular stress and carcinogenic events. Diverse combinations of these very complex processes may lead to intracellular instability. Many aneuploid cells die, because severe intracellular instability is generally very harmful to living organisms. In addition to that, the immune system can also recognize and eliminate abnormal cells. However, highly adaptive karyotypes will be able to survive even in a harsh and unfriendly microenvironment by outcompeting other cells for nutrients. Their selective advantage is due to amplification of some oncogenes, which are frequently overexpressed in human cancers. Simultaneously, one or more crucial tumor suppressor genes will be inactivated through mutation or deletion. As a result, rapidly growing aggressive tumorigenic phenotypes will evolve through malignant transformations, which can be represented by a set of common properties, the well-known hallmarks of cancer, as first proposed by Hanahan and Weinberg in the year 2000 and later amended by others. Highly resistant mutated lineages will proliferate, grow, and gain territory by clonal expansion. These processes inevitably lead to the formation of macroscopically observable tumors in the body. Most solid tumors are very heterogeneous in nature and show increased intracellular instability over time. Chromosomal imbalance and massive aneuploidy are also associated with chemotherapeutic drug resistance. It is becoming increasingly evident that the genomic instability strongly influences clinical outcome. Several studies have shown that expressions of selected genes implicated in chromosomal destabilization process could be used for diagnostic and prognostic purposes in the clinical practice. After having seen the importance of intracellular instability, we will now discuss metabolic flexibility. The growth of a malignant tumor is highly regulated by its microenvironment. Besides the availability of nutrients and oxygen, which are essential for cell survival, interactions with various elements of the extracellular matrix also play a fundamental role. As we have seen, tumorigenesis is greatly influenced by intrinsic cellular diversity due to intracellular instability. Manifold combinations of these parameters lead to large intratumor heterogeneity, which gives cancer cells the ability to stay alive even under dissimilar circumstances. In extremely adverse environments, part of the cells die. However, resistant cells that acquired high adaptability and metabolic flexibility during tumor evolution can reorganize their genomic content and survive even drastically harsh hypoxic situations by entering into reversible cell cycle arrest called quiescence. This state is characterized by a decreased metabolic rate and largely reduced biosynthesis. Highly energetic processes are stopped. Quiescent cells are not growing, not proliferating, and produce only low amounts of reactive oxygen and nitrogen species. When nutrients are chronically sparse, an autophagic signaling program can be activated whereby damaged organelles and other non-essential cytoplasmic components are degraded to provide energy to the starving cell. Tumoral cells can develop sophisticated metabolic mechanisms to survive in the quiescent state for months or even years. However, cancer cells located in close proximity to blood vessels are more likely to rapidly grow and proliferate. In a worsening environment, they are also able to go quiescent due to their high metabolic flexibility. This transition is reversible. Under favorable nutritional conditions, dormant cells can be activated by metabolic readjustment and re-enter into the cell cycle. Thus, metabolic flexibility allows tumoral cells to switch from one fuel source to another depending on external situations and regulate their energy consumption between wide ranges. The nutrient utilization of proliferating cells increases dramatically due mainly to overexpressed glucose and glutamine transporters and diverse metabolic enzymes. Nutrients are consumed swiftly to support cellular functions and advance anabolic synthesis of cellular building blocks, leading to increased tumor growth. To stimulate further growth, malignant cells produce an elevated amount of growth factors. Due to the high metabolic rate, more heat is generated, and some energy can be expended for the production of inflammatory cytokines. These effects lead to chronic inflammation, fueling further tumor evolution through a positive feedback loop. Another very significant consequence of the hyperactive catabolization of glucose and glutamine is the increased production of proteases as well as oxidative and acidic compounds. The flexible and versatile metabolic machinery facilitates the successful upregulation of the tumor defense system to either directly block the attack of immune cells or hide and escape immunosurveillance. Likewise, they are able to survive hypoxia by generating new blood vessels in a process called neoangiogenesis. As pointed out by Jim Watson, Michael Lisanti, and others, high concentration of oxidative molecules can kill cancer cells too. To defend themselves, they apply sophisticated antioxidant and coordinated transport strategies to reduce radical concentrations inside and increase it outside. This causes forceful oxidative stress in neighboring cells, leading eventually to their death or malignant transformation. The intracellular accumulation of hydrogen ions disturbs the sensitive pH homeostasis, and a number of mechanisms are activated accordingly, as can be seen in this illustration taken from the recent work of Neri and Sepuran. This creates a transmembrane reversal of the intracellular to extracellular pH gradient. Quite contrary to normal cells, cancer cells have a higher, slightly alkaline intracellular pH and a lower, slightly acidic extracellular pH. A key protein in promoting an acidic microenvironment in tumors is the sodium protein exchanger isoform 1. In aggressive neoplasms, it is highly overexpressed, constitutively active, promotes invasiveness, and confers poor patient prognosis. The lower external pH reduces the viability of nearby stromal cells and provides an evolutionary advantage for adaptive tumoral cells through transmembrane pH reversal. This finally results in an acid-mediated tumor invasion of the parenchyma, a concept advocated by Gatenby, Gillies, and many other researchers. Tumor invasion is further advanced not just by oxidative reversal and inflammatory processes, but with the degradation and remodeling of the extracellular matrix through secreted proteases as well. These immensely complex processes are mediated by thousands of signaling proteins, including several key players shown here. And now we can turn our attention to the third pillar, cytoskeletal plasticity. The cytoskeleton has three main structural components: actin filaments, intermediate filaments, and microtubules. The cytoskeleton has an essential role in the regulation of various cellular processes, including morphology, growth, and movement of the malignant cells. The spatial and temporal dynamics of the cytoskeleton is regulated by hundreds of proteins. This biological system is so staggeringly complex that the possible number of interactions among the signaling proteins greatly outnumbers the estimated number of stars in the entire known universe. Nevertheless, recent advances in molecular imaging by optical nanoscopy, atomic force microscopy, and single-cell video microscopy have enabled us to visualize key intracellular elements and processes, as well as to gain a deeper insight into the operation of the cytoskeletal machinery. With the help of high-tech bioinformatics and super-resolution real-time live cell imaging, we are now beginning to understand the key mechanisms connecting cytoskeletal plasticity with tumor evolution and metastasis formation. During mitotic cell rounding, dividing cells undergo radical shape change, as you can see in this brief video in the central part of the screen. A growing number of recent publications confirm that massive dysregulation of different cytoskeleton-associated proteins, in large part due to processes related to intracellular instability, result in aggressive and motile phenotypes. Microtubules take part in many intracellular processes, including cytoskeleton remodeling, vesicle transport, and cell division. Their precisely coordinated actions are absolutely essential for successful mitosis. Based on this fact, a number of microtubule-targeting chemotherapeutic drugs have been developed and are in clinical use, including taxanes, Winke alkaloids, and epautilones. To facilitate passage through dense tissues, cancer cells use focal adhesions and various actin-rich plasma membrane protrusions like lamellipodia, filopodia, and invadopodia. Rho family members and other signaling proteins stimulate actin filament growth, which pushes the cell membrane forward. This figure, which is taken from the excellent paper by Reskin, Cardone, and Targundi, outlines the role of diverse proteins in the dynamics of invadopodia. Notice that the natrium hydrogen exchanger NAG1 is located at the tip and contributes largely to the cleavage of the extracellular matrix through hydrogen ion efflux. A number of studies have indicated that the cytoskeleton linker protein ezerin contributes to metastasis formation in a variety of cancer types. Focal adhesions through integrin signaling mediate proliferation and cell migration. The actin-related protein 2 and 3 complex is responsible for the branching of the actin filament, has matrix degrading capacity, and plays a critical role in protrusion formation. Coffulin has been found in several studies to be significantly upregulated in metastatic tumor cells. The scaffolding protein cortactin is a central element in cytoskeleton restructuring. Its overexpression has been correlated with poor survival prognosis in these cancer types. This picture illustrates well that depending on the structure and stiffness of the local microenvironment, cancer cells can utilize different morphologies and formations to move forward. Highly metastatic cancer cells have been reported to display cannibalistic behavior. During nutrient scarcity, migrating tumoral cells may attack, engulf, and digest other cancerous or normal cells. Tumor cannibalism can also significantly contribute to immune evasion and drug resistance. This process requires high cytoskeletal plasticity and often leads to ploidy increase and intracellular instability, which again, as we discussed in the first part of this video, promotes aberrant cancerous proliferation. In this video, we tried to briefly summarize and visualize how cancer develops to be a fatal disease. The large intracellular instability of malignant cells leads to metabolic flexibility and cytoskeletal plasticity through Darwinian selection. The weak die, the strong get stronger. During this process, tumor cells become highly aggressive, invade their neighbors, and travel into distant organs to form new colonies. This metastatic development is usually the main cause of cancer-related deaths. We mentioned at the beginning, and would like to emphasize again, there are a myriad of other characteristics, interactions, and pathways involved in carcinogenesis. We have listed here several interesting and actively investigated aspects. Unfortunately, there is a large discrepancy between the great number of theoretical concepts and the modest clinical success rate. One reason for that might be that there are a lot of cause and consequence correlations that we are not aware of yet. Hopefully, this black hole of cancer will be more and more illuminated by innovative research in the coming years. But for now, our personal approach to cancer is a very humble one, because we realize what a formidable task it is to cure patients with metastasis. Keeping that in mind, in our next video we will nevertheless discuss possible clinical implications of the 3 pillars of advanced solid cancers.